Hello, I am Dr. Hughes, and I'm very excited to share the work we've been doing to better understand the genetics affecting pergolide response in horses with pituitary pars intermediate dysfunction, or PPID. This disease, also known as equine Cushing syndrome, occurs when a portion of the pituitary gland that is located at the base of the brain overgrows. This overgrowth then leads to excessive hormone production, and a variety of clinical signs can be appreciated, including a long, shaggy hair coat that fails to shed, loss of muscling along the horse's top line, and laminitis, a painful condition that affects the hoof and can cause loss of use or even loss of life. PPID is a very important disease in horses, as it affects up to 25% of horses above the age of 15, leading to many affected animals undergoing treatment in the U.S. each year. A challenge currently facing veterinarians is that despite the importance of this disease and much continued research, it remains difficult to accurately diagnose these horses, especially early in disease. There is also a current treatment for this condition, a drug called pergolide, that helps to mitigate clinical signs. However, this treatment is lifelong for the horse and does not cure the condition. This treatment requires dedicated owners and veterinarians that will continually work together to manage these horses. One challenge with pergolide is that it tends to be effective in less than 75% of cases, and a portion of these horses will suffer adverse effects that can limit our ability to treat them. This has caused us to consider, why are these horses failing treatment, and how could we better predict how a horse is going to respond to drug therapy? One potential answer is the difference in genetics between individuals, and our project aims to determine what is the genetic contribution to pergolide response in horses. In order to answer this question, researchers in our lab have begun enrolling horses with PPID that have a broad spectrum of response, as well as variable adverse effects present when on therapy. Data on their drug therapy, doses, clinical responses, and details of these adverse effects are being collected through a survey. Veterinary records are then being obtained, including lab results, and horses' individual blood samples are being submitted for isolation of DNA. Once this DNA is isolated from the enrolled horses, it gets sequenced for previously prioritized mutations so that we can determine the horse's individual genetic code at each site. Differences between these sites are then identified and studied to determine the association with pergolide response. For example, potentially the location of a C at this base becomes associated to a strong response to drug therapy, or the location of a T may contribute to the need for altered dosing, or an A increase the likelihood that a horse becomes anorexic on treatment and has to stop the drug. This example is quite simplified, and in reality, thousands of these comparisons are being performed across the genomes of multiple horses by harnessing the power of computers. This analysis will help us to identify genetic differences that can be used to better predict responses to therapy and then guide future research projects. Being able to better predict when a horse will require a higher dose, when pergolide may not work at all, or potentially when a combination protocol would be more effective, would save owners time, money, and frustration when treating affected horses. As advanced vet care and management strategies have improved, a larger number of our horses are living longer, making this disease even more common. Improving our ability to effectively treat horses with PPID has the potential to be extremely impactful to the equine industry. Please visit our website to learn more about this study. We are thankful for you listening today and very thankful for the funding that makes this project possible.